Dr. Muhammad Salleh Rofiee did his PhD in I Promise under the supervision of Professor Dr. Tehlaike and Professor Dr. Muhammad Zaki, and he successfully graduated in 2016. And for your information, Dr. Salleh also is one of the scholarship recipient for the program Scientist dan Penyelidik Muda, or also known as PSPM in UiTM. His expertise is none other than metabolomic, and as far as I'm concerned, there are not so much researchers actually venture in this field. So currently, Dr. Salleh is one of the I Promise Fellow, and he is also the person in charge for the bioanalytical unit. So without further ado, I would like to welcome and invite Dr. Muhammad Salleh to deliver his talk.
Okay, thank you, Dr. Tengku, for kind introduction. So let me start my presentation. Wait 4 minutes. Good morning to everyone. First of all, I would like to thank to the organizer, the Office of Deputy Vice Chancellor, Research and Innovation for giving me this opportunity to be part of this webinar series 2.0. I also would like to thank all the participants for joining this event. The title of my presentation today is The Frontier in Metabolomic, Discovery of Marker. In the first part of my presentation, I would like to share about the metabolomics in general, and the second part of my presentation, I will share about our experience in I Promise doing metabolomic. What is metabolomic? Metabolomic is a field of life science research that use high throughput technology such as LCMS, GCMS, NMR to identify and characterize all the small molecule or metabolite in the cell, tissue, or organism. Some people also call it study of metabolome. What is small molecule or metabolite? It is any organic molecule detectable in the cell, tissue, or organism with a molecular weight less than 1,000 Dalton. It is including endogenous and exogenous molecules such as food metabolite, drug and drug metabolite, bacterial metabolite, and many more. Just as genomic in the study of DNA and genetic information within the cell, and transcriptomic is the study of RNA and differences in mRNA expression. Protein is the study of protein. Metabolomic is the study of substrate and produce metabolism with influence both genetic and environmental factor. This is the metabolomic, which is the study of the metabolite in our body. When we talk about the study of small molecule, the first paper was by Robinson and Pauling, entitled The Quantitative Analysis of Urine Vapor and Breath by gas liquid partition chromatography. This paper was published in the Proceeding of National Academy of Science in 1971. This is the first paper on metabolomic, even though at that time not called metabolomic. As I mentioned before, metabolomic is the study of small molecule. The name of metabolomic was coined in the late of 1990s. In 1998, the term of metabolome was introduced by S.G. Oliver and his partner. Their finding was published in Trend in Biotechnology in that year. So basically, they study on the metabolome of the yeast. When we search in PubMed using keyword metabolomic, we can see the trend of publication was increased from the year 1998 when the first metabolomics term was coined by O.G. Oliver until more than 7,000 publications in 2019. Why now researchers or scientists look at the metabolomic? Not all the changes or abnormalities detected in genome or transcriptome may be causing abnormality or disease. Example, silent mutation. Similarly, not all enzyme and protein products detected via proteomics are functional. Also, they do not take into account environmental influence occurring at the later stage. Metabolomics also can use to monitor changes in genome or measure effect of downregulation or upregulation of specific gene transcript. Metabolites also are ultimate result of cellular pathway. That's why now researchers look into the metabolomics. So, metabolomics basically can be divided into two approaches. First is untargeted metabolomics, or people call it global metabolomics. The goal of this untargeted metabolomics is to measure as many metabolites as possible from biological sample without bias in order to generate metabolic profile of a sample. This is the general workflow for global metabolomic analysis. The second approach is targeted metabolomics. This approach typically used to answer specific biochemical questions in pharmacokinetic study of drug metabolism, as well as for measuring the influence of therapeutic or genetic modification on the specific enzyme. So this is the general workflow for targeted metabolomic analysis. Now, this slide shows general workflow for global and targeted metabolomics. It starts from sample collection, sample treatment, and also processing. After we collect the sample, we need to extract out the metabolite or small molecule in the sample. After that, we need to use chromatography technique to separate all the extracted small molecule and detect using mass detectors such as mass spectrometry. Usually, researchers use high throughput instruments such as LC-MS, GC-MS, and NMR. Of course, after we get the data from these high throughput instruments, such as LC-MS, GC-MS, or NMR, we need to do analysis and validation using other omic approach. When we talk about the sample for metabolomic study, it can be from cell culture, which is suspension and adherent cell media, tissue, or feces, and the most popular among researchers is biofluid, such as serum, plasma, blood, urine, and gland secretion. How we want to collect and handle the sample is very important. It is because small molecule or metabolite inside the sample is very sensitive. It may change within a second. If we fail to handle the sample properly, maybe the data or result that we get is not represent the real situation. So, in this case, all the biological sample collected for metabolic analysis require careful sample handling, special requirement for their diet, physical activities, and also patient validation. All the sample must be placed all in minus 80 degree or dry ice if we do in the other place and transport for our lab. all the samples should be remain in minus 80 degrees until the extraction process. Also, the sample should not be thawed period to extraction process. Basically, the standard sample volume for biofluid is around 0.1 to 0.5 mil, and for tissue, standard sample weight is around 100 milligram. This is the general step how we want to extract the metabolite from the biofluid. In this case, we use around 150 microliter of serum. Then after that, we add 150 microliter of distilled water and vortex to mix them, and use ACN to precipitate the protein. This step was done twice, basically to make sure all the biofluid is clear from the protein. After that, we dry the sample and reconstitute with the mobile phase before we inject to the system. Now, this is the extraction technique for tissue sample. So in this case, we use 100 milligram of tissue, or around 1 times 10 power of 7 number of cells. Then we add the around 300 microliter distilled water, and we homogenize the cell and or the tissue in ice cold condition to make sure all the metabolites are can be protected from degrade. After that, we use methanol to precipitate the protein, and to extract out the non-polar compound, we use chloroform and vortex it again. After that, we combine the aqueous part and also the organic part, and use the concentrator to dry the sample, and before we inject, we reconstitute with the 30 microliter of mobile phase. After we extract out the metabolite or small molecule, we need to separate them using either gas chromatography, capillary electrophoresis, high-performance liquid chromatography, or ultra-performance liquid chromatography. From this graph, we can see the most sensitive instrument detect small molecule is LCMS. We can see here from this graph, LCMS is more sensitive to detect the small molecule. But each instrument have their own advantage. For example, NMR can give structural information, but not for LCMS and GCMS. So after we get the data in the format of chromatography, we need to do baseline correction, peak alignment, compound extraction, normalization, and deconvolution using software such as MassHunter Qualitative and also XCMS. Then we need to do statistical analysis to make sure only the significant metabolite is further for identification. In this statistical analysis, we usually do the univariate analysis, unsupervised multivariate analysis, supervised multivariate analysis, and multi-way method. All these statistical analysis use software such as Mass Profiler Professional and also Metabolomics. To identify the compound, researchers and scientists refer to the spectral database and library. For example, HMDB and also HMDB and methylene database for human metabolite, food database for FoodDB is for food database. To study the metabolic pathway involved, a few software can be used. like Cytoscape, MedPA, and BioCYC. For the validation of the annotated compound, targeted MSMS or other omic approach need to be done. Okay. Now, we move to the second part of my presentation, which is research on metabolomic and I-Promise. In I-Promise, we use LCMS-Q-TOF-based metabolomic study. One of our projects is to study the hepatoprotective effect of Muntinja calabula extract against carbon tetrachloride-induced liver injury in rat using metabolomic approach. This study has been done and published in the Journal of Ethnopharmacology. In this study, we use rat as a model. Basically, we administered the rat with different dose of Muntinja calabura for 7 days, and after that, we induced hepatotoxic to the rat using toxicant called carbon tetrachloride. After 24 hours in induction, we collect the blood and use the serum for metabolomic analysis. Before we proceed with LCMS Q-TOF analysis, the protein precipitation was done to make sure the sample were clean from protein. This is important. If our sample is not cleaned properly, the protein will stuck into our column. For the data analysis, we use several software such as Mesh Hunter analysis for data conversion from the .d data that we get from the LCMS Q-TOF to .cef, and we use Mesh Profiler Professional to do the statistical analysis, and for ID browser identification, we use Methylene database here. And for the pathway analysis, you use open source software which is MedPA, and biomarker identification we use our software and we validate all the finding using a targeted MSMS approach. From the finding, we can see that the normal rat, the metabolite for normal rat is significantly different from the CCl4. We can see the cluster of the normal metabolite and also the carbon tetrachloride metabolite. So basically, this is the animal that we induce hepatotoxic using CCl4. So this is the animal without any inducer. So after that, we also compare the animal which give pre-treated with Muntinja calabura extract for 7 days, and also the animal that did not give any pre-treatment. So we can see the separation in terms of metabolite for both group of animal. Another PCA show that when we compare the Muntinja the animal with that give pre-treatment with Muntinja calabura and the normal animal, we can see the metabolite. In terms of metabolite, there is no difference because all the metabolites are clustering together. Same like the when we compare with the silymarin. Basically, silymarin people use when as a standard drug for the hepatotoxicity study. Okay, so even it is different, but we can see there is no clear separation between both group, between the pre-treatment with Muntinja and also the silymarin group. Okay. After that, we do the biomarker. We find the biomarker. Based on the profile, we found that xanthosine, this form metabolite, can be used as a biomarker. All these four metabolites involved in the purine metabolism, primary bile acid metabolism, and also arachidonic acid metabolism. So, as a conclusion, this study has successfully isolated four major pathways involved in the hepatoprotective effect of Muntinjakalabura against carbon tetrachloride-induced liver injury using LCMSQ-12 metabolic approach. And the involvement of arachidonic acid and purine metabolism in hepatoprotective has not been reported previously and may provide a new therapeutic target and option for treatment of liver injury. So in this study, we managed to find the new pathway that can be used as therapeutic target for the treatment injury which is not reported before this. Okay, so from the metabolomic, yes, we get this new information from that. In another study, we use metabolomic to find new biomarker for myocardial infarction. This finding was published in the Journal of Clinical Bioinformatics. So the title of this paper is Metabolomic and Partial Least Square Discriminant Analysis to Predict History of Myocardial Infarction of Self-Claimed Healthy Subject, Validity and Feasibility of Clinical Practice. Okay. Basically, this study used serum as a sample. Firstly, we profile the metabolite from myocardial infarction patient in healthy volunteer. After we find the significant metabolite, we process using univariate analysis, ROC, univariate ROC curve analysis to find biomarker. From this, we developed the prediction model. After successfully developed the model, we did the phenotype prediction of orang asli, whether they are healthy or have a cardiac risk. Okay, whether they are healthy or cardiac risk. So, this is the finding. In this case, most of the metabolites which are expressed among myocardial infarction patients and healthy were lipid. So, based on this metabolite differently expressed among patient and healthy volunteer. So, from the finding, we can see the difference in terms of metabolite profile between patient and healthy volunteer. After we developed the prediction model based on the biomarker that we were identified earlier, we predict the healthy status of orang asli. We predict why we choose orang asli, because they are claimed they are a healthy person. From the result, we found that some of them are not healthy and have a risk to get myocardial infection. So, they are very interesting findings here, because most of them self-claim they are very healthy, but they have a risk to get the myocardial infarction without their known. In this study, the model was used for predict CVD risk among orang asli, and some of the orang asli self-claim health status were shown have metabolite profile like a patient. So from the finding, this show that metabolomic is useful as an alternative to the routine biochemistry test. Besides the two projects that I mentioned earlier, we also use metabolomic study in breast cancer patients. In this study, we tried to compare the different expressed metabolite between healthy control and breast cancer patients and identify the potential markers in breast cancer patients. The second objective is to profile and identify the differently expressed metabolite in estrogen receptor negative and estrogen receptor positive and the metabolite that were differently expressed in different grade of breast cancer. In this study, we use plasma and also tissue as a sample. Based on the result, we found that 22 metabolites were differently expressed in the plasma of breast cancer patient and when compared to healthy volunteers. So we can see here the blue one is the breast cancer patient and the red one is the healthy volunteer. We can see the separation here. That means the metabolite from the healthy is different when we compare to the breast cancer patient. Same as the previous project on orang asli, we also developed the model based on the biomarker identified earlier for the breast cancer patient, and we use blind sample to test the model. So this is basically the the model that we developed. So in the left-hand side is the healthy, and the right-hand side is the patient. When we add the unknown sample basically, so we can see here the red color, the left hand side, the red color in left hand side is show that the person have a risk of breast to get breast cancer, while the right hand side show there are no risk in getting breast cancer. But in the between, so let's say like this dot, and this dot have a potential to get the very high potential to get the breast cancer patient, even though they are in the healthy side. When we look into the metabolite from the tissue sample, we found that the icosanoate metabolite trend lower in the level as tumor grade increase. So that means when the grade of the tumor increase, the level of icosanoate was lower. Okay. Same like spermine was increased in in estrogen receptor positive and upregulated in grade 3 breast cancer when compared to grade 1 and grade 2 breast cancer. It was known that the majority of the estrogen receptor negative tumor are in grade 3. So as a conclusion, Metabolomics managed to identify difference in metabolism pathway between the normal and breast cancer sample. Pathway are identified to be related to energy and cell proliferation and protein. Metabolite profile in estrogen receptor positive and negative samples show differently regulated metabolites such as spermine. We also found that icosanoate compound and spermine were found to be differently regulated in tumor with different grade, like grade 1, grade 2, and grade 3. So, based on the example that I have give, metabolomic approach can be used to find a new biomarker, disease prediction, and used in toxicology study and also drug discovery. And many more other applications can be used for using metabolomic approach, such as nutrient metabolomic, disease diagnostic development based on the disease prediction, based on the biomarker that we find using metabolomic approach. So metabolomic also can be used to use in personalized medicine and also human metabolome study. Before I end my presentation, we in iPromise provide a service for qualitative mass spectrometry and also quantitative mass spectrometry services. Okay.
So thank you so much, Dr. Salleh, for that very insightful presentation, and it really helped us in understanding the metabolomy even more better. So I believe that some of the audience here, participants here, would like to ask questions, to you regarding your presentations or regarding some of the metabolomic studies that have been conducted by your team. So now I open the floor for the participants to ask the questions. You have two choices. Number one, whether you can unmute your mic and strictly talk to Dr. Salleh, or if you feel a little bit shy cat, so you can type your questions in the chat box, and I'll try my best to assist Dr. Salleh in order to read all your questions. So you can ask now. We have approximately 20 minutes. Any question from the floors? And I just would like to give any questions?
Assalamualaikum Dr. Salleh and Dr. Tengku.
Waalaikumsalam, Prof.
Nur Hadiani here.
Yeah.
Okay. I just want to ask a very basic question to Dr. Salleh. Can you just elaborate a little bit more about the process of identifying the biomarkers, because that's the title of your presentation, because from your multivariate analysis, your PCAs and so on, and how do you get to the biomarker identification? And when you say about new biomarkers, what do you mean by new there? Okay, because I suppose all these compounds, metabolites are actually known metabolites. So what do you really mean by new markers? Okay, all right, that's it.
Thank you, Prof. So for the biomarker identification, basically from the metabolite that we identify using ID browser identification from the database like HMDB, methylene database. Then after that, we go to the other software which is AUROC. That from the that software we know the specificity and sensitivity of the biomarker. Then after that we have a few criteria like the sensitivity mostly like to 1, and then after that we need to validate the biomarker. We need to validate the biomarker using MSMS approach. So after that then we proceed with the clinical trial lah. So for the another question, the new biomarker means that there's no reported yet the compound has been involved in the certain disease. Yeah, I think I answered the question.
Okay, thank you, Dr. Salleh.
Welcome, Prof.
Okay, Dr. Salleh, there is actually one question from Nur Hartini Dolhaji. I hope that I pronounce your name correctly, So, Assalamualaikum, Doctor. As metabolomic helps in disease prediction, how far is this application being used as biomarker in drug development in Malaysia, I guess? Yeah, so that's the question from Nur Hartini Dol Haji.
Okay, so as I mentioned before, from the biomarker we can predict the we can do the disease prediction. So if we know the disease will earlier, so from that we can prevent it from the in term of macam apa, we can predict earlier, then we can help to treat the disease earlier. Yeah.
A new question from Dr. Mashani Muhammad. So, may I know whether validation using MSMS is really necessary, or is it possible to validate using gene or protein expressions, or both? Thanks.
Yeah, for validation using MSMS, basically we want to confirm the that is the exact compound. So yeah, it's a good if we can validate the biomarker also using gene or protein expression or both. It is better if we can do the MSMS gene expression and also protein expression for the validation process. Yeah.
All right, is there any questions? Okay, questions, yeah, from Joseph. Okay, how accurate is biomarker to predict a disease like cancer? You mentioned orang asli said they are healthy, but you found some have biomarkers for heart problems. How big is this population that are involved? Thank you.
Okay. So how accurate the biomarker predict disease like cancer? So basically, this from the orang asli, that is just we build a model. So we build a model for cardio CVD problem. Then after that, we add the orang asli. This orang asli claim their self is a healthy. Okay. So we add them into the model and we can see uh, some of them are not healthy and have a risk to get the CVD. So we get back to their healthy report and we can like the blood profile everything, and we can see some of them that fall into the high risk with CVD have a potential to get the CVD. Yeah. Basically, the population is around 120 people for orang asli. Yeah. And for the CVD is around 100 people involved in the study. Yeah.
All right, while waiting for other questions to be pop up in the chat box, yeah, I would like to just remind you to don't forget, yeah, to sign your attendance, which has been posted by the secretariat in the chat box. And another quick announcement is actually we still have another 2 webinar series from our I Promise Fellow, which will be on next Monday and next Wednesday. And the title for the next Monday presentation from one of our fellow is From Microbes to Microbiome. And the other webinar series on Wednesday next week will be on molecular diagnosis in clinical parasitology, a game-changing approach. So don't forget, yeah, that we have another 2 webinar series from our fellows, I Promise fellows. All right, Norafardia Izati, say thank you for sharing your knowledge. Yeah, most welcome. Any questions from the participants? Salam, kenapa saya tak boleh masuk link pendaftaran tu ya? I think maybe Secretariat can answer your question. And if you are interested in doing some of the metabolomic study, you are feel free to come to our I Promise facilities, yeah, to discuss with Dr. Salleh and other research team members, yeah. So, but make sure please make an appointment first, right? So, All right. Any justification? I think last question would be from Ahmad Kamaramli. Any justifications of this on current COVID-19?
So far we have not done any experiment on COVID-19. So I think for COVID-19, we can use metabolomics to understand what actually happened in our body based on the small molecule. Based on the small what actually COVID-19 did in our body, and our body respond to the COVID-19 based on the metabolomic. From that, we can know the pathway of how the COVID-19 involved in the body. Yeah, that is my comment on the question.
So, no more questions from the participants. All right, I think it is the time now for us to leave the webinar series. Yeah. So again, thank you so much for all the participants who are joining from 10 a.m. Yeah, ada yang masuk awal lagi. So thank you so much for your attention. Thank you so much, Dr. Salleh. And again, thank you so much for the organizer